Hello, welcome to the next in a series of review clips designed to assist you in preparing for your Biology 3201 public examination. In this clip, we'll be taking a more in-depth look at population genetics and Hardy-Weinberg principles. With population genetics, we are simply studying the genes in the population, how they may or may not change over time. In other words, has evolution occurred based on genetic distributions in the population? If there's a shift in the gene pool of a population, we know that evolution is indeed happening. We define a population as being a localized group occupying a particular area. And remember, the gene pool is the total of all genes contained within that population. One means by which we can verify that populations are indeed evolving is by applying the Hardy-Weinberg Law. This law was independently discovered by Godfrey Hardy and Wilhelm Weinberg. What it does is it explains how Mendelian segregation indeed influences allelic and genetic frequencies within a population. There are a number of assumptions that are made in order to verify and apply the Hardy-Weinberg Law. First, we assume that the population is large. This avoids genetic drift, which is a change in genetic frequency due to chance. Secondly, mating is a random process as it pertains to traits you are studying. Third, we are excluding the impact of natural selection again for the traits under study. Fourth, no mutations occurring. And fifth, no migrations occurring. With these assumptions in place, we can determine and calculate the genetic frequency of a population that is indeed in equilibrium. If these assumptions are met, the population is deemed to be in this genetic stasis or equilibrium. Based on this, we can expect two predictions. First, the allele frequencies are not changing, and after a generation of random mating, the frequencies will remain in the following proportions. P squared is the frequency of the homozygous dominant genotype. Two PQ is the frequency of the heterozygous genotype. And Q squared represents the frequency of the homozygous recessive genotype. In short, P represents the frequency of the dominant gamete. Q represents the frequency of the recessive allele. And we add them together to form P squared plus 2PQ plus Q squared equals 1, or 100% of the population. As well, P plus Q equals 1, where P equals the frequency of the dominant allele and Q the frequency of the recessive allele. Remember, look at these assumptions in specific time now. Firstly, that the population is infinitely large. Right away, well, this assumption is unrealistic. But a large population will mathematically function similar to an infinitely large one. Finite populations with rare mutations, rare migrants, and weak selection, in general, however, do indeed fit Hardy-Weinberg proportions. The second assumption, that mating is random. Now, very few orders mate randomly for all traits in all loci. But the Hardy-Weinberg situation will apply to any locus for which mating occurs randomly, even if the mating is non-random for others. Third, no natural selection. Fourth, no mutation. And fifth, no migration. In short, we want the gene pool to be closed to the addition or subtraction of new alleles. Like random mating, this applies only to the locus under study. So let's take a look at an example. Take humans, Drosophila, even for X-linked traits. In females, the Hardy-Weinberg frequencies are the same for any locus, p squared plus 2pq plus q squared equals 1. In males, the genotypic frequencies are the same as allele frequencies, p plus q. Now we know that recessive X-linked traits are indeed more common among males, but the proportions remain. Let's take a look at some sample questions from past public examinations. In this question, as posed, 9% of individuals are homozygous dominant for tongue rolling. What percentage are heterozygous for tongue rolling? Remember, the two forms you need to be applying are p plus q equals 1, p squared plus 2pq plus q squared equals 1. You are told that 9% are homozygous dominant. If 9% are homozygous dominant, p squared equals 9%, which equals 0.09. To calculate what p is, or the frequency of the dominant allele, we take the square root of both sides. P then would equal the square root of 0.09, which equals 0.3. As p plus q equals 1, q equals 1 minus p, which equals 1 minus 0.3, which equals 0.7. You are looking for the individuals that are heterozygous. We can calculate this by looking at 2pq, which represents heterozygous. So 2 times 0.3 times 0.7 equals 2pq, which equals your heterozygous individuals, which equals 0.42. 0.42 is 42%. Another question is posed on a past public examination. Two alleles, A and little a, are in Hardy-Weinberg equilibrium within a population. If A is 0.7, what percentage show the dominant trait? Remember, the dominant trait will be shown by those individuals that are homozygous dominant and those individuals which are heterozygous. Unless you were told otherwise, you would assume complete dominance. If big A, or P, equals 0.7, Q must equal 1 minus 0.7, which equals 0.3. Remember, P plus Q equals 1. To calculate the individuals that are homozygous plus those that are heterozygous, we need to add p squared plus 2pq equals, in this case I'll call it x because that is what we are looking for. So 0.7 squared plus 2 times 0.7 times 0.3 equals X. 0.49 represents our homozygous dominant. Our heterozygous individuals is 0.42, which equals 0.91, or 91%. Another sample question, in this case a constructive response question, asks you about the first human mission to Mars was a tremendous success, theoretically. A self-sustaining colony established consisting of 3 females and 6 males, all of reproductive age. Give 2 reasons as to why this population would not remain in genetic equilibrium according to Hardy-Weinberg. In other words, why would Hardy-Weinberg principles not apply over the long term? The answers provided would be any 2 of the following: a small population size, non-random mating, the possibility of mutations, the possibility of natural selection. All of those items, all of those things would not allow the population to remain in genetic equilibrium. The population is small, mating is not random. This question is posed, again, asks you about the Hardy-Weinberg calculation. You're told that the frequency of a recessive allele is 0.2. What percentage would be expected to show the dominant trait? Remember, P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. You are told that Q, the recessive allele, is 0.2. So P would equal 1 minus 0.20, which equals 0.80. You are looking for those that show the dominant trait, or your homozygous dominant, plus your heterozygous individuals. A quick way to calculate this is that if you know the number that show the recessive trait, the remainder will show the dominant. As Q is 0.2, Q squared equals those that show recessive, which equals 0.20 squared, which equals 0.04. If 4% of your population shows the recessive, the remainder must show the dominant, or 96% would show the dominant form. The correct answer is indeed D, 96%. This question is posed indicates that a recessive trait appears in 81% of the population. What percentage of the population will be heterozygous? In this case, again, remembering that P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1, we can work from the 81% that shows the recessive to determine Q, and subsequently P. If Q squared equals 0.81, Q must equal the square root of 0.81, or 0.9. If Q is 0.9, P equals 1 minus Q, which equals 1 minus 0.9, which of course equals 0.1. You are looking for the heterozygous individuals. Remember, heterozygous is 2PQ. So the numbers that are heterozygous would be 2 times 0.9 times 0.1, which equals 2 times 0.9 times 0.1 equals 0.18. correct answer is 0.18 or 18%. And I also present the following. In a population of squirrels that is in Hardy-Weinberg equilibrium, 91% are gray and 9% are black. If black is recessive, what percentage of the population is homozygous dominant for this trait? If black is recessive and black is 9% of the population, remember, as P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1, we can work out the numbers or values for P. Q squared, the recessive, equals 0.09. Q then would equal the square root of 0.09 or 0.3. P would equal 1 minus 0.3, which equals 0.7. Heterozygous, as you recall, is represented by 2PQ. So therefore, the heterozygous is 2 times P, or 0.7, times Q, or 0.3. This equals 0.42, or 42%. The correct answer would be B, 42%. Thank you for your attention. I hope you found this learning clip to be helpful.